Namaste everyone, and a very warm welcome to today's Masterclass Explainer. I'm absolutely thrilled you've joined us because, well, today we are going to put on our detective hats. We're diving headfirst into what I consider the absolute greatest biological mystery of the 20th century. It's a true, microscopic whodunit.
For decades, you see, the brightest scientific minds were completely stumped by one profound question. What exactly is the secret of life? Which molecule actually holds the blueprint of heredity? Was it protein? Proteins are complex, they're everywhere in the cell. Or was it DNA, a molecule that, frankly, many initially dismissed as just too simple? Well, let's examine the evidence together.
Here's the roadmap for our investigation today. We'll start with the initial mystery of heredity, then dig into the legendary Hershey Chase and Mendelssohn-Stahl experiments, see how scientists actually photographed the blueprint, and finally wrap up with the monumental mapping of the human genome. We have a lot of ground to cover, so let's get started.
Chapter 1. The Mystery of Heredity. The Search for the Secret.
The story really kicks off back in 1871. Imagine this, a scientist named Frederick Griffith ingeniously isolates a substance he calls nuclein. And where does he get it? From salmon sperm, of all things. Then we jump to 1928. Frederick Griffith stumbles upon this transforming principle, something that could actually turn completely harmless bacteria into deadly ones. It was shocking. By 1944, Oswald Avery, Colin MacLeod, and Maclyn McCarty really ruffled some feathers in the scientific establishment. They boldly suggested this transforming principle was actually DNA, not protein. But you know how it is. Science demands absolute, undeniable proof, which brings us to the year 1952.
Chapter 2. The Hershey Chase experiment. Protein or DNA?
This brings us to two of my absolute favorite scientific detectives, Alfred Hershey and Martha Chase. They came up with a genuinely brilliant strategy. To figure out if it was DNA or protein acting as the genetic culprit being passed down, they zoomed in on the chemistry. It's a beautiful fundamental difference, really. DNA contains phosphorus, but absolutely no sulfur. Protein, on the flip side, is packed with sulfur, but has barely any phosphorus. This simple chemical distinction, that was their key to picking the lock.
But every good detective story needs a villain, right? Meet the T2 bacteriophage. It looks exactly like a tiny lunar lander, doesn't it? It's actually a virus, and its sole purpose in life is to infect E. coli bacteria. The outer coat of this virus is made entirely of protein, but tucked away inside its geometric head is DNA. It lands on the bacteria, injects its genetic material, and totally hijacks the poor bacteria to build new viruses. The million-dollar question was, what exactly was it injecting down that tube, the protein or the DNA?
So, how on earth do you track something so unbelievably small? Hershey and Chase used radioactive isotopes. Think of them as microscopic GPS trackers. They cooked up one batch of viruses using radioactive sulfur, so that tagged the protein code. Then they made a completely separate batch with radioactive phosphorus, which tagged the DNA inside. They let these microscopic invaders attack the bacteria. And then, this is the best part, to detach the empty virus shells from the outside of the bacteria, they used a blender. Yes, a standard everyday kitchen blender. Just imagine, profound Nobel Prize-winning biology being done with kitchen appliances. After a quick blend, they checked inside the bacterial cells.
And the results were absolutely definitive. No doubt about it. The radioactive sulfur, it all stayed outside the cell, stuck in those empty protein shells. But the radioactive phosphorus, it had completely moved inside the bacteria. This beautifully simple, elegant experiment proved it once and for all. DNA is the genetic material. The great scientific debate was finally, decisively settled.
Chapter 3. Photographing the blueprint. The blueprint revealed.
Okay, so now we know the true culprit is DNA. But what does this master blueprint actually look like? Well, to see something that incredibly tiny, you need a very, very special kind of camera. Enter X-ray fiber diffraction. Over at King's College London, around May 1952, two brilliant researchers, Rosalind Franklin and her PhD student Raymond Gosling, were mastering this exact technique. They were literally firing X-rays at incredibly thin fibers of DNA to capture the shadow patterns they made as the rays bounced off. And that painstaking work gave us the legendary Photo 51. It was quite literally Gosling's 51st photograph, and it gave an exceptionally clear diffraction pattern of the B form of DNA. That distinct, beautiful cross shape right there.
That was the crucial, undeniable piece of evidence. It was the exact clue scientists needed to finally visualize the tightly wound double helix. A single photograph that quite literally changed our understanding of the natural world.
Chapter 4. The Messelson-Stahl Experiment. How DNA replicates.
So the structure of DNA was finally solved. Amazing. But instantly, a new, massive puzzle reared its head. How does this double helix actually copy itself to pass information on to the next generation? Back in the 1950s, scientists were fiercely debating three main theories. First, the semi-conservative model, championed by Watson and Crick, where the two strands unzip and each one builds a brand new half. Second, the dispersive model by Max Delbruck. He suggested the DNA sort of chops itself up into bits, copies them, and glues it all back together. And third, the conservative model, which proposed that a completely new double helix is formed from scratch, leaving the original parent DNA totally intact. So which one was it? To solve this, Matthew Messelson and Franklin Stahl at Caltech designed what many scientists still call the most beautiful experiment in all of biology. Just listen to these incredibly elegant steps. First, they grew E. coli bacteria in an environment full of heavy nitrogen, so all the parent DNA became artificially heavy. Next, they moved those bacteria into an environment with normal, light nitrogen. As the bacteria divided and multiplied, the scientists extracted the DNA and spun it at super high speeds in a centrifuge. Now, because heavy DNA naturally sinks lower than light DNA, this was literally like color-coding the parent and daughter strands. They could see exactly where every piece ended up.
And the result was breathtaking. When they photographed the bands under ultraviolet light after exactly one generation, the DNA bands sat perfectly in the middle. It was a hybrid. Exactly half heavy, half light. This instantly and definitively proved the semi-conservative model. The DNA unwinds and every single parent strand acts as a precise template to build a newly replicated daughter strand. Such elegant, precise, and profoundly beautiful scientific logic.
Chapter 5. The Human Genome Project. Mapping our DNA. This brings us to our final chapter. Fast forward to 1990. The monumental, almost unbelievable goal? To map the complete set of human DNA, to read the entire book of life. Because the human DNA strand is so unimaginably long, scientists had to use something called the hierarchical shotgun approach. Think of it like taking a massive, million-page encyclopedia, ripping it up into individual pages, so thousands of people all over the world can read a different page at the exact same time. They broke the genome into large, manageable chunks, let bacteria copy them, and then sequenced them all separately. It was a painstaking, heroic global effort.
And what was the final, breathtaking result? 3.1 billion. That is the staggering number of base pairs in the human genome. 3.1 billion microscopic biological letters making up the instruction manual to build you, me, and every human being. And you know what is truly beautiful to remember about that massive number? The vast, vast majority of that sequence is completely identical across all humans on this earth. Biologically speaking, we are so much more alike than we are different.
Which leaves us with a final thought to ponder as we wrap up today's Masterclass Explainer. What mysteries are still hidden within those 3.1 billion base pairs? We finally have the map in our hands, yes, but we are truly only just beginning to understand the language it's written in. What future genetic discoveries are waiting right around the corner for you, the next generation of scientific detectives? Think about that. Class dismissed, my friends. Until next time, keep questioning and always keep exploring.